Hi guys, so I'm going to be teaching you about mitosis, meiosis, cancers, and cells today, and we're going to focus on meiosis first, and we're kind of so I actually missed questions number 4, 6, 7, 10, number 21, 24, 38, and 39 from version C test, and so I'm going to begin by talking about meiosis. And basically, meiosis is a reproduction phase of cell division, and basically, it's making gametes, which is sperm and egg cells, for reproduction. And so basically, in interphase 1, not much is happening at the time. Basically, the chromosomes are just doing its normal function. The nucleus is just in its normal function. However, then in prophase 1, the chromosomes pair up. And so basically what happens is crossing over begins to happen with the chromosomes. So crossing over basically means that some of the genes are transferred to a different chromosome for like genetic variety. And then we get to metaphase 1, and this is where everything starts to get interesting. So this, in prophase 1, the DNA was copied from interphase, and so once all that happens and everything crossing over happens and everything, we get to metaphase 1, which is basically where the chromosome pairs line up in the middle. So and they usually line up based on size, so we're just gonna kind of have different sized chromosomes. And so basically what is happening is that they're lining up, they're getting ready to split. So the spindle fibers and then here are the centrioles at the end. And then we'll get to anaphase 1, which is where the chromosomes are pulled away by the spindle fibers. And so you'll kind of see stuff like this happening. I'm trying to draw it as clearly as I can. And they're being pulled away by the spindle fibers. The centrioles are at the end, so this is just the whole thing. And then you get to telophase 1, which is cytokinesis splits them into 2 new cells. And so you're in anaphase 1, they're being split apart, and then they kind of form into their own. And so cytoplasm is splitting with cytokinesis right here, splitting them into 2 groups, and you get 2 new cells. And that's the end of phase 1 for meiosis. But then new things start to occur. And you get to prophase 2. And this is kind of where the chromosome and spindles start to form in both cells. So they have basically prophase 2, they are in 2 different cells, like we saw before. They're in their 2 different cells. And then metaphase 2 happens. Which is where both the cells line up in the middle, except it's in single file this time instead of in pairs. So all the chromosomes are lined up in the middle. And they're single file. And this is where the spindle fibers start to form again. You get the centrioles at the end. And then we'll get to anaphase 2. Which is basically where the chromatids, sister chromatids, get pulled away by the spindle fibers. So, in both the cells, this is happening. So we have, these are the chromosomes, sister chromatids being separated. Spindle fibers are pulling them away. And this is happening over here as well. You get the spindle fibers pulling them away too, centrioles at the end. And then you will get to telophase 2. And cytokinesis again occurs, and this is where 4 new cells form. So basically this splits, and so all of this becomes one cell, all of this becomes another cell, all of this becomes one cell, all of this becomes another cell. So it'll end up looking like this. And then that's when you get the 4 new daughter cells. And usually at the end, this is where 46, you get 46 chromosomes in each, even though you start out with that. Or no, you get 23. You get 23 chromosomes into each, even though you start out with 46, because you're making the sperm and egg cells and making gametes. And so that's how reproduction for meiosis happens. And now we are going to talk about mitosis. In mitosis, this is where you're actually making not a genetic variety like we did in meiosis, but in mitosis, this is where we make 2 identical cells, and so this is needed for repair, replacement, growth, etc., etc. And so we will start with interphase, which is basically not much is happening. It's just a normal phase of the cell cycle that the cell is just doing its job. So you have nucleus, and you have your chromatin all in there, which is your DNA. And then you get to prophase. And this is where each strand duplicates itself. So you will end up getting all these chromosomes pairing up. And the nucleus is still there, it's just condensing and thickening. And then once we get to metaphase is where the nucleus disappears. And this is basically where all of the chromosomes line up in the middle. So they all line up in the middle, and then you have your spindle fibers forming. And the centrioles at the end. And then you get to anaphase, which is where the sister chromatids separate. And so you have your one, and then all the sister chromatids are separating. You have your spindle fibers, and centrioles at the end. Then you get to telophase. And this is basically where the new nucleus is forming and to make two new daughter cells. And so cytokinesis is occurring and splitting the cytoplasm. And forms the two new cells. And they are identical cells. And that's how you get mitosis. This can be deadly for cancer cells because uncontrolled mitosis is cancer and can lead to rapid cancer growth. So basically, a tumor is a cluster of cells going through uncontrolled mitosis, and malignant tumors are cells that do migrate to other parts of the body, but benign tumors are cells that do not migrate to other parts of the body. So when mitosis is uncontrolled and is rapidly spreading the cancer cells, that's when mitosis can be deathly and not be repairing and helping with growth. Hi guys, so today we're going to be learning about the characteristics of living things, and the questions that I got wrong on the test for version C were number 36, 37, and 40A. And basically a really good way to help remember the characteristics of living things is an acronym called RODRIGS. So we are going to start with the first one. For R of RODRIGS is reproduction. And a good way to remember that is reproduction, like cradling a new baby. You're making a new organism and you're making a new cell, essentially. And so another good way to remember reproduction is through genetic material and new DNA. Basically, there's a new organism. And then another characteristic of a living thing is order and organization. And so a good way to remember this is by just tapping on yourself. This is a good body gesture. So order and organization, just knowing that there's an order to all organisms. Like we have organs, we have organelles, we have all different parts of our body that are organized to make us who we are. And that can be like cells, tissues, chemical balances, pretty much like any system that has an order or structure in your body makes you a living thing. Another thing is adaptation. And so one way you can remember this, fighting hands. I remember this as survival of the fittest. So getting up your fighting hands, adaptation. So this is you are able to survive and reproduce in the environment that you're put in. And the characteristics that you have allow you to survive and reproduce. And then the D in road rigs is development and differentiation. And so this is a good way to know basically that you're figuring out what you want to be when you grow up. And so a good way to remember this is going height. And so just like knowing that you're maturing and you're developing and different parts of you are changing. Regulation is a good way to remember this is to go like this because it you're regulating your body. So I think of it as like tanning. You're regulating your body's pretty much regulating and having homeostasis and knowing what it's functioning and it's able to survive in the same state and maintain the same internal conditions. So that's R, regulation, in the first part, or the second part for regs. And then E for regs is energy, and one way to remember this is eating. So all living things get energy from another source, and so that can be food, that can be sunlight, that can be water, but basically all living things need something in order to get energy to be living. And then growth, I think of this as growing, so like larger. Basically this means that you're increasing in size and your number of cells. So you're increasing in your size and your number of cells. You're essentially just getting bigger, and that's different from development and differentiation because your cells and your tissues are maturing during your development, and so that means it's functioning differently as you continue to grow, and grow means like the physical, you can see yourself growing. These cells are, there's more cells that are being produced, etc., etc. And then the last one of road rigs, the S is sensitivity, and so I think of this as shivering. And so shivering, sensitive, you're sensitive to the things that are put around you, and so that your body reacts, or whatever living organism reacts to the environment it's put in. And so that means, like for plants, some of them will drop their leaves responding to different environments. Some will lean towards the light if they see sunlight. There's different ways that they will respond to their environment. So remembering that reproduction, cradling the baby, order and organization, adaptation, development and differentiation, regulation, energy, growth, and sensitivity. So just remember those, and you'll have a great time with remembering characteristics of living things. Thank you. Hi guys, so today we're going to be talking about photosynthesis and the general life cycle of a plant. And the numbers I missed on the exam for this question, or this topic, are questions number 27, number 42B, 29, and 41A. So today we're going to start with the general life cycle of the plant, and that starts out with the seed. And the seed is germinating, and how it gets that is sunlight, air, moisture start the process of seed germination. And so from there, the seed grows into a plant. And during this stage, the seed detects the pull of gravity and sends root down with gravity and shoots up against the gravity. So it's detecting light and nutrients, and it's kind of knowing, oh, hey, gravity is a thing right now. And so the next one, or the next stage, is the plant turns into a flower. And so during this, after it has its leaves and roots, and it's no longer dependent on food supply from the seed, it starts to make its own food. And this is a process, photosynthesis is when it starts making its own food. So this is also the stage where the plant matures and flowers, which you see here, which contains its reproductive organs. The stamens produce pollen and the pistils produce ovals. So we're going to kind of show you what the process of photosynthesis looks like. So here's a plant, and there's the sun right there. And basically what is happening is first the plant is taking in carbon dioxide. And then through this, the plant is also bringing up water from its roots. And the sunlight, the light energy, makes chlorophyll, and so it's getting its light energy. The molecule plant used to capture light energy is chlorophyll, and so through this light energy, chlorophyll captures the light energy. And then through this, oxygen is released, and then sugars are either stored or released as well for other organisms, and it creates its own food. So getting back to that, sorry, I just lost my flower. So getting back to that, we are at the flowering stage. And basically after this is when the flower turns into a fruit. So during this stage, pollination is occurring and the transfer of pollen from the anther to stigma. And after pollination, the flower transforms into fruit with the seeds inside. So we'll see that. And this is kind of the stage where the seed disperses either by wind, water, insects, birds, and it's kind of traveling along, and the seed is then planted and going through the same cycle again. And through that, the plant begins to die. And then all the other seeds that were dispersed, they start the cycle again. And that is the general plant cycle. And that is pretty much all we are talking about today. Perfect. See you guys later.